If you've been listening to the Business of Biotech podcast for a while now, you'll recall that Erin Harris has joined me to co-host a few episodes. Erin's my friend, colleague, and chief editor over at cellandgene.com, and she just recently launched a podcast of her own. It's aptly named Cell and Gene, the podcast. And if you're working in the cell and gene space, you should give it a listen. It's a collection of interviews with the industry and academic leaders moving the space forward. And you can find it at cellandgene.com or anywhere you listen to podcasts. Cell and Gene, the podcast. Check it out. This is the Business of Biotech. I'm Matt Pillar, and I'm about to get out of my depth and into yours with Dr. Gene Lee, VP of Technical Development at AltruBio. After earning his BS in biology from Carnegie Mellon, Dr. Lee was awarded a PhD in microbiology from New York University and did a postdoctoral fellowship at the Dana-Farber Cancer Institute and Harvard Medical School. He built a biotech career on that platform, serving as principal research scientist at Genetics Institute in Wyeth, director of cell line development at Persivia, and senior director and global head of protein and cell sciences and U.S. site head, discovery and development technologies at EMD Serono before joining AltruBio in April 2021. Today, we're talking about CMC developability assessments and more specifically, how leveraging them during the discovery phase of novel biologics speeds up entry into phase one. But first, let's get to know Dr. Lee a little bit better. Dr. Lee, hi, welcome to the show.
Hi, Matt. Thanks for having me.
It's our pleasure. I'm thrilled you could be here. And as I said, I'm out of my depth, so I'll lean heavily into your expertise as we pursue these questions. But I'm looking forward to learning a lot today. But before we jump into that, I want to spend a little bit of time learning a little bit more about you. You made this transition from academia to industry relatively early in your career. What was the motivation for the application of your biology training to drug development?
Well, it might come off as a little bit corny, but I've always wanted to help people. I've always wanted to do something that would have an impact on people and more specifically patient health. I knew I wanted to be a scientist from when I was pretty young. My dad was a biochemist, and I spent a lot of time as a kid just hanging out in his lab. And because my dad was in academia, he was a scientist, I had always imagined that I would follow in his footsteps and work in an academic lab as well. And I was always sort of gravitating towards those laboratories that were working on, the way that the body protects against diseases, so the way the body keeps itself healthy. Mm-hmm. And so I worked in laboratories and on topics that were really related to host cell immunity. So as a PhD student, I worked on interferon and tumor necrosis factors. So these are cytokines which are involved in immunity. And as a postdoc, I worked on aspects of the immune system as well. But I never really felt terribly satisfied working in an academic setting. I just didn't feel like my work was having the kind of impact that I was looking for. And so I began looking at opportunities in industry. Now, this was sort of in the late 90s when I was finishing up my postdoc. And I would say at that time, the biotech industry was still pretty young, not a whole lot of companies, certainly not the kind of ecosystem that you and I live in today. And so I wasn't, well, let's put it this way. I was very naive about the opportunities which were available to a scientist who wanted to work in industry. Fortunately, I did my postdoc, as you said, up at Harvard Medical School, so in the Boston, Cambridge area. And there were a few young companies that were beginning to emerge. And long story short, I found myself at Genetics Institute, very lucky to do so, in an organization that was really focused on development. Mm-hmm. There I was able to just kind of use the skills that I'd learned as a PhD student and as a postdoc to really apply sort of the basic fundamental science to bringing drugs to market. And, I think something there clicked. This is the way that I could have the biggest impact in applying what I know and what I love to really helping people.
Yeah.
The rest is history.
Yeah. Well, it seems apropos that you end up at a company called AltruBio, given your altruistic
There you go.
intent. So another transition that's noteworthy, looking at your CV, is the transition from big bio to emerging biotech. So EMD Serono is pretty good sized company. The transition to AltruBio, being a clinical stage company, what was the motivation for that move?
Yeah, so I've been in the industry for well over 20 years now. And in the end, I wanted to do even more to really help bring drugs to people who need them. It was a hard move to make. It was a tough leap to make from EMD Serono to join AltruBio. I had a really successful career there. As you mentioned, I was a global head of a department that was bringing drugs into the clinic. I was a site head and really able to stretch my knowledge and my experience into fields which were a little bit less familiar to me, and interacting with a lot of people, a lot of really brilliant scientists. But it was a larger organization. And so as such, a lot of the decisions that needed to be made had to go through several layers of governance and committees. We often had to make very tough decisions about which asset to move forward, which asset to kill, because obviously, we didn't have an unlimited budget to move everything forward. Sure. And that's tough, right? You work on a project for years, only to have it killed because it's decided that it's less interesting than another molecule that the organization might be working on. I think at AltruBio, given its size and its really intense focus, I really have the opportunity, or I feel like I have the opportunity to more directly link the science to bringing meaningful drugs to patients, and really linking the science to strategic decisions, moving things that we think make sense forward.
Cool.
I mean, it really goes back to my initial motivations of wanting to help people. I think at AltruBio and a small company like AltruBio, I just have the chance to make more meaningful and impactful decisions every day. I should add as well that joining AltruBio was made a little bit easier because I know Judy from my days at Wyeth Biopharma. We were colleagues. I guess 20 years ago, working up in Andover, Massachusetts, and I think the opportunity to work with her again was the clincher.
Yeah. Did you say 20 years ago?
Yeah, I think it was about 20 years ago.
You guys, you and Judy are both aging very well. It helps though when you make transitions like that to have the confidence in someone like Judy, someone like Dr. Chow, who you've worked with before and been able to develop that confidence in her ability to lead. I totally get that. I've made similar career choices where I find myself back with people that were great leaders and good to work with in the first place. So very nice. I want to start transitioning into this more technical conversation around developability assessments, but I want to start at a bit of a higher level. Given that AltruBio is a clinical stage company, before we get specifically to CMC developability assessments, I want to discuss phase 1 and just how important a milestone that is to emerging biopharma companies, and the obstacles that companies face getting there, and that developability I'm sure is one. So, what are when you look at the obstacles, the challenges that a company faces as it prepares for, leads up to phase 1? What kind of concerns you the most?
Yeah, it's a really great question, Matt. And I think obviously that the answer is multidimensional, right? So to get a drug successfully into phase 1, you have to look at clearly the science behind the concept. It has to be solid. And there has to be a business case for doing it as well. It's a big investment for any company to go into phase one. So is your molecule going to be competitive? Is it differentiated enough from other molecules which are either already in the clinic or in the preclinical stages of development? But you're talking to a CMC guy, so I look at phase one readiness, through the lens of CMC developability. And so that's a technical aspect then, which is a little bit distinct from the sort of core science and the scientific rationale for bringing a molecule forward.
So define that. That's a beautiful segue. Define that for us. What's define CMC developability for those folks who aren't necessarily coming at it from a technical perspective like you.
Sure. I guess in a nutshell, you could sum it up by saying, can you make the damn thing? So,
okay. Yeah.
Right. You might have a great idea, a brilliant idea of how to cure a disease. You're going to make an antibody that targets something on a tumor cell or a T cell, let's just say. And that by doing that, you're going to affect the biology of the disease in a way that you think has a good chance of treating the disease. Well, so then you have to start thinking about what will that molecule look like? How do you design that molecule? And really, that's the first step in developability. It really starts at concept generation. Mm-hmm. You think about the molecule almost like, thinking about building a car. So if you're working at Tesla, for example, and you want to build a brand new car, what are the design elements there that will lead to a successful product in the end? So once you have that design, you often then need to prototype. You build several versions of those early designs, see if they work, put them out on the racetrack, see what kind of range it has, this sort of thing. And not all designs are going to work well for whatever reason. You might have a great idea about what the molecule should look like or what the car should look like, and it just doesn't work for whatever reason. And even if the concept is great, you've picked the right target, it's a disease that has an unmet need. If you can't make the molecule, if you can't manufacture it, it's just not going to go anywhere. And in my experience, it's better to think about those things early before you've really invested too much time, too much energy into a project to only find out that you can't make it. That's expensive. It's heartbreaking. I mentioned to you, at a larger organization, you often have to make those choices. Mm-hmm. Sometimes those choices are because of CMC considerations. The molecule just isn't able to be made either at all, and that's rare, or made efficiently. And that's also an important factor because, a molecule which you can just kind of barely squeak out in a manufacturing setting is likely going to be expensive to make. It's going to have a long development program. And those costs inevitably get passed on to the patient and to payers. And, I think we're all very aware of the sort of rising costs of drug care.
Yeah. How early? So we're talking about we're talking about executing these developability assessments early, right? Mm-hmm. Discovery phase. But it occurs to me that there may be a too early, right? It might, there may be a point in the discovery phase that's just too early to assess the developability because maybe we haven't, progressed that far. And if we kill it too early, we'll make a mistake that we regret and maybe never know we made a mistake, right? Yeah. So I guess, I'll simplify back to the simple question. How early is too early?
I get the question. And I think it can be addressed by thinking of developability perhaps in a phased approach, right? So in my opinion, there isn't a too early. Okay. There might be investing too much too early component of your question. So, you certainly wouldn't want to throw the kitchen sink, the mother of all developability assessments, at the earliest stage of your discovery efforts, because you also don't want to squash innovation or ingenuity very early on. You want to give those ideas a chance to breathe, a chance to really see what they can do from a functional perspective in your discovery laboratories.
Mhm.
And so an early developability assessment might be essentially just prototyping a large number of potential designs and just dropping the ones that are less promising than others. So you're not killing the program, you're just sort of putting aside, perhaps for now, those designs which are less developable than others, but you keep the program moving. Later on, as the program matures and you're beginning to gain a little bit of momentum and steam, then you can begin applying a more mature, more, let's say, process relevant assessment. Some people call this a manufacturability assessment. In this case, you're really prototyping the molecule in a process which resembles what you will use on the manufacturing floor. So it's really a good predictor then of how your molecule will behave in a manufacturing setting. Going back to the Tesla analogy, you can do the early assessments maybe in your garage. That's an early kind of assessment, but at some point you're going to want to put it out on the racetrack to see how it runs, and have it circle the track all day to see how reliable it is and how durable it is. And that sort of later assessment is not something you want to do early on before you've had a chance to really flesh out the ideas.
Sure. Are those are those assessments are they fairly standardized? Are they proprietary approaches? From company to company. Is there a framework? Is there guidance?
They are pretty standardized, and I think that comes from the fact that when we're talking about biologics, by and large, over the past 10, 15, maybe 20 years, when you say biologics, you mean monoclonal antibodies. Right? Mm-hmm. So that's a very standard sort of molecular structure, and with that comes a standard way of manufacturing them, of processing them. And so too, with developability assessments, you can evaluate monoclonal antibodies in what we call a platform process, see how well they are expressed in a recombinant cell line, see how well they behave. So some antibodies have a tendency to clump together, aggregate, and those, generally speaking, will not be developable in the long run. But you can assess that pretty early on. And it is predictive then of developability and manufacturability issues later. Now, I will say that over the last several years, there's been increased interest in other kinds of molecules. So not monoclonal antibodies, but things like bispecific antibodies or Fc fusion proteins. These are a little bit more complex than your standard antibody. And they have, I would say, by and large, a greater chance of not being developable. And so you really have to look at it early on to make sure that you're not, placing all your bets on something that, just you can't make it in the end.
The Business of Biotech is brought to you in partnership with Cytiva. Together, we're committed to helping the leaders of new and emerging biopharma companies navigate the financial, organizational, human resources, and regulatory waters you'll encounter on your way from discovery to the clinic and beyond. Check out a host of useful resources for biotech leaders at Cytiva's Emerging Biotech Accelerator at cytivalifesciences.com/emerging biotech. That's CYTIVA lifesciences.com/emerging biotech. So on the flip side of that, you pointed out a couple of things that might point to a lack of developability. What are you looking for in the assessment that bode well for developability?
Yeah, high expression level is kind of the cure for a lot of ills. So if you can express your molecule at a very high level in your process. You can have a crappy purification process, and you're still going to be okay because you'll have enough material in the end. So, I would say that's the number one thing. The other thing, again, is the aggregation of the molecule, but the clumpiness of the molecule. That also is a good predictor of how it will behave in purification and formulation. I think there's a product quality aspect to that as well. I would say if you can look at those two things early on, you're in good shape. Obviously, there are some other quality attributes that you want to take a look at as well. But these fall into the very sort of standard analytical methods that companies who are working on biologics will utilize every day anyway.
Okay. What resources are required to execute or conduct a developability assessment? I still want to kind of get my mind around like what that looks like in practice in the day-to-day operations of the company. So we're looking at a discovery phase, developability assessment as discovery is going on. Is Dr. Lee and his team toiling away in a lab, executing a developability assessment on its own and reporting back to the team, or does everything pause while these assessments are happening? Give me a better idea how that plays out.
Most discovery laboratories will have some kind of protein expression group. You need that kind of group in order to supply material to all the research teams to do all their preclinical studies, their animal studies, what have you. Mm-hmm. And of course, the protein that's being supplied should be of, let's say, sufficient quality. You don't want to provide the teams with material where you'll have questions, about the data and the results. So laboratories which are equipped to do this sort of early protein supply, early research supply, I would say are already pretty well equipped to do a developability assessment. They're the first set of eyes on how the molecule expresses. They will have to purify the molecule. And so they will also begin to gain experience about how the protein behaves. Is it aggregating? Does it tend to fall out of solution under different salt conditions? They will run analytical assays to look at the quality of the molecule that they're sharing, with their research teams. And so they will see, what is the percent monomer, on a size exclusion chromatograph, for example. So these labs are already well equipped to do these early developability assessments. Now, I think that to do it really well, the sort of early research labs should have an understanding, at least, of what the manufacturing process looks like. And that can either be through interactions and conversations with an internal manufacturing and development organization. Of course, this is really true only in larger companies that have that internal capability. Or it can come through the experience of the scientists and the leadership in a smaller organization who are really much more attuned to what the needs in development and manufacturing are. I mentioned to you that it's a good idea to check the developability or manufacturability in the context of the manufacturing platform. So this is racing the Tesla on the racetrack. So having that conversation with either your internal manufacturing group or, if need be, with an external CDMO about what their platform looks like, will be a big help, because then you can test your molecule in the context of whatever that platform is. And it just gives you greater confidence that once the research team is ready to hand over their project over to manufacturing, they'll be able to just to run. And the fact of the matter is, at that point, once you've decided to manufacture, the clock is ticking. You're really sort of on the path to IND. You don't want there to be any delays in moving that product forward. You want to spend as little time in process development to get into phase one at that point.
Mm-hmm. Does the molecule ever So the developability assessment of the molecule, does that ever inform the manufacturing process or platform, or is it always the other way around? The way that you describe it, Dr. Lee, it kind of sounds like, we have to make sure that we can develop this molecule within the parameters or the guardrails of the platform with which we're subscribing to manufacture, whether it's internal or with an outsourced partner. Does it ever kind of work the other way where you go, look, like we think this thing is developable? We've done the assessment. We think there's an opportunity here, but it's going to require some maybe heavy lifting or reworking of our manufacturing capacity.
What you're describing, I think, is what the state of the industry was when I first started. So when I started at Genetics Institute, monoclonal antibodies, really weren't on the big stage yet. Platform processes didn't really exist. So at that time, companies like Genetics Institute, like Amgen, like Genentech, were really working on replacement factors. So we were working on blood clotting factors like factor 8, factor 9, EPO, insulin. These are the kind of molecules that the early biotech companies were working on. And each of those molecules just, they behave differently. There's nothing similar from one to the other. And so if you wanted to bring those kind of molecules into the clinic, you'd have to completely develop, your process from scratch in order to handle the sort of unique properties of each of those molecules. So, that was kind of the paradigm back then. When the industry really shifted to monoclonal antibodies, and today, you've probably seen all the data about what the top 20 biotech products are, and it's largely dominated by monoclonal antibodies. Why? Because they're very similar to one another, and it's easy to set up a platform process. A platform process meaning it should fit in the same kind of cell culture system. It should fit in the same kind of purification system, one molecule to the next. And so you can really develop a portfolio around monoclonal antibodies to move a lot of different products into the clinic. And then once that started happening, people really started to look at process efficiencies and facility utilization. How can we move these molecules into the clinic faster and faster, beat the competition, lower the cost of goods, lower the overall cost of manufacturing. And so now that mindset has dominated the thinking in the industry. Now we're seeing a shift away from monoclonal antibodies, as I mentioned, Fc fusion proteins and immunocytokines, things like this. And while they do share a certain similarity to antibodies, they're also quite different, not only from an antibody, but also from each other. And there is a tendency, I think, still to try to make them fit in the antibody platform. I mean, by God, we've spent so much money in investing in a facility to handle an antibody process. Might as well, find a molecule that'll fit that.
Yeah.
But sometimes it's just not possible. You have a really great idea. You've built a great molecule, and as you say, Matt, we think we can develop a process around this. We just don't have that process yet. And so in those situations, then I think there has to be a conversation with the development and manufacturing organizations to see what the best strategy moving forward is.
Yeah, excellent. What's and that I was going to lead into that question. Like what you mentioned, the monoclonal early days of MABs, when I asked that very naive question about being able to fit the molecule to a new manufacturing process. I was curious if we were in those kind of early Wild West days as it relates to Fc fusions and bispecific antibodies and so on. So thanks for getting ahead of that one for me. I want to shift gears a little bit and talk about the way that you transfer knowledge. The transfer of knowledge taken from a developability assessment to make sure that data is transferred effectively and perhaps even informs process decision making and scale up and so on. Does it? Yeah. Are there formal systems in place for that transfer of knowledge?
Probably for some organizations, the answer is yes. And that's probably more likely in organizations that can support both a discovery research organization and in internal manufacturing, right? Because then you have a very clear path of communication, from one side to the other. And I think that's critical. So obviously you want to transfer information from discovery research into development. This is the molecule we're working on. We're handing over that molecule and all the knowledge that we've gained over to you guys to develop a process. But there also needs to be the communication and transfer of knowledge backwards as well. So the process development folk can develop a very robust platform for making recombinant molecules, for purifying the molecules, and they can transfer that knowledge back to the research team so that they can evaluate the molecules in that context. I think that's important. I'm not sure that exists all the time with every organization that I've come across, but I think really the best ones try to integrate those two teams in some sort of CMC developability kind of sub-team. Now, for smaller organizations that don't have internal manufacturing capabilities, obviously they will need to outsource those activities to the CDMO. But there again, I think there are opportunities for knowledgeable CMC experts from both sides to really take a look at the overall package before the formal execution of a project, to really anticipate what the challenges might be, what we think might be the best path forward, whether a platform process could be implemented. Likely that's going to be the cheapest and often the quickest way to the clinic. And if we have to deviate from the platform, what are the best options to consider? I guess in a nutshell, it really comes down to communication in both directions.
Yeah. Okay. What haven't I asked you about CMC developability assessments that I should have?
Yeah.
As I said, I'm the first guy to admit that I'm stepping into this conversation going, I don't even know really what that means. So I'm hoping to learn from Dr. Lee. So help me out here. What should I have asked you if you're coming at it from the perspective of the leader, tech leader of an emerging biopharma?
Well, Matt, you wouldn't be the first person to tell me that they don't understand developability. And I think actually that's part of the problem, the issue. So, I worked in a discovery organization. I've also worked in a development organization. But in discovery, I think the focus is really mostly around functionality of the molecule, right? So what is the binding affinity? What does the target engagement look like for that antibody? And CMC is usually either, a secondary consideration, or they haven't even heard about, what CMC is. But I think it's important to really take a holistic approach to developing a drug, to designing a drug, and you really need to take the time. So my advice really primarily to discovery organizations would be to take the time to do a proper developability assessment, because not doing it will often lead to spectacular failures in the end. A molecule which looks great on paper, maybe works great in the laboratory, but you can't make it, and so the project is dead. And that's I think the absolute worst outcome for a discovery organization, particularly for a smaller organization that might be really banking a lot on the success or failure of that molecule in the clinic. You want to be able to at least get to the stage where you can demonstrate proof of concept in the clinic. But to do that, you have to make the damn thing. So take the time to do a proper developability assessment.
Yeah, I like it. So I want to, before we wrap up here, I just want to point out that not too long ago, just a couple episodes before this, I interviewed I presume she's your boss, your CEO, Dr. Judy Chow at AltruBio. If I bring that up because if folks among our listenership are interested in learning more about the company the pipeline that you're actually exercising these developability assessment chops on, the actual pipeline, listen to that episode. It's not even numbered yet because it hasn't been released, but look for Dr. Judy Chow in that episode, and you'll learn more about the work that AltruBio is doing. It's important work and diverse. So I congratulate you on the successes you've seen and the work you're doing. And I'm super honored that you were able to join us, Dr. Lee. Thank you for your time.
Yeah, this was a lot of fun.
I hope so. I'm not afraid to ask naive questions. And I think that's why people like to tune into this podcast. They're like, let's listen to that guy who asks naive questions, because they yield transparent responses, right? And I appreciate you for that.
Yeah, no, it was fun. And hopefully I was able to explain things in a way that you and the audience can understand.
For sure. I feel smarter. So thank you. So that's AltruBio VP of Technical Development, Dr. Gene Lee. I'm Matt Pillar, and this is the Business of Biotech. We're produced by Bioprocess Online in partnership with Cytiva. If you're not already subscribed to my newsletter, please visit me at bioprocessonline.com and sign up for it. And check out Cytiva's Biotech Accelerator at cytivalifesciences.com/emerging biotech, where you'll find a host of resources to aid you on your journey. Finally, if you're benefiting from the conversations you're hearing on the Business of Biotech, please hit that subscribe button and give us a 5-star review. In the meantime, thanks for listening.